It's my honor to introduce my advisor, James Dahlman. Dr. Dahlman did his Ph.D. at MIT in the HST program under Dan Anderson and Dr. Robert Langer. He then did his postdoc with Feng Zheng, a pioneer in the CRISPR field, and created some gene editing tools that allow for orthogonal gene editing. So that'll be my intro, and I'll turn over to James.
Great, thanks. Hey everybody, good morning. How you guys doing? Good. So Corey, thanks for the introduction. No pressure. I told him if he messed up, it only cost him 2 years on his thesis, so no big deal. You're actually going to be seeing Corey's picture a little bit later today, along with one of his colleagues, Kalina. They've done a great job. A lot of the data that you're going to see in the second half of the talk, which is, in all honesty, I think the more interesting data, have been generated by Corey and Kalina. So I'd like to thank them ahead of time. I'm sorry this is... well, hopefully it isn't too stretched out. Can you guys see this? This should be all right. The title of this presentation is not a typo. The first time I gave this talk was about 6 weeks ago. And the first question I got I thought was perfect. Somebody raised their hand and they said basically, hey, you have a typo in your title. This should be in vitro. And I said, no, that's not actually a typo. And the point of this talk is hopefully to convince you guys that we can actually test hundreds of nanoparticles in a single experiment now directly in vivo in any combination of animal models that you might like. And so if you do a few experiments in a row, you can get into the thousands. Okay, so just to set the framework here a little bit, my lab works on drug delivery, and I think what we really do is we work on technology development and apply some molecular biology tricks in order to improve drug delivery systems. So we take my background, which is in chemical engineering and nanoparticle design, as well as genomics and CRISPR-Cas9 and sequencing and all these sort of broad institute type technologies, and we have them talk to one another. And I really like this space a lot. But again, to set the framework, at the end of the day, we're interested in drug delivery. And although the technologies we're going to describe later can be applied to either oral or local delivery, the majority of the data that you're going to see are related to systemic delivery. So this is just IV injections into mice right now. We focus on drug delivery of genetic drugs. I'm particularly enamored of RNA. And there's a reason for this. These are just 3 papers that have come out this year in the New England Journal of Medicine, which I think is a key trait here. We're not talking about scientific papers anymore in scientific journals. Instead, we're talking about clinical papers. And there is a coming revolution in siRNA, and it's starting to get here. So these are all patient data that have been generated with siRNA drugs. All of them target the liver. But this is happening now. There's likely to be an approval for an siRNA-based drug next year, very, very likely. And then probably 1 or 2 the year after that, and then the flood's going to start to come. So RNA-based drugs for gene silencing are becoming a real thing in the clinic. This is one data set I like to show, and I had to update it. This is just a year old. But what you see here, I just want to demonstrate the fact that these siRNA drugs are real. This is patient data. This one will highlight patient data. And this is after subcutaneous injection. Okay, so this is an siRNA drug that's put into a little syringe, then you inject it like an insulin shot, basically. And what you see is this is gene expression on the y-axis, and this is time in days on the x-axis. And for those of you, just to set this framework, for those of you who don't know about siRNA drugs, siRNAs turn off genes. So the cool thing about siRNA drugs is that you can turn off this gene, and then by switching around the RNA sequence, turn off that gene. You can turn off any protein coding gene in the genome that you would like. So what you should see is after you give an siRNA drug, the gene expression should drop. And that's exactly what you see. So what you see is robust gene expression, robust gene silencing, upwards of 75 to 90%. And the amazing thing here, and that's why I like to highlight here, you get 75% gene silencing for 6 months. Okay, so as far as I know, the best silencing we've seen in people is a subcutaneous injection at a pretty reasonable dose. And you can get silencing for, I think, 4 or 6 months after 1 injection. The reason why this is important is because if you compare this to an antibody-based therapy, this makes RNAi competitive now for antibody-based targets, at least in the liver. This is liver data. Okay. So when you look at the clinical field of RNAi, this is what the field looks like to me. And I don't mean this to be offensive. I think I'm allowed to say this because I work on non-liver delivery. So this isn't diminishing or minimizing any of my own work or the work done by my colleagues. But if you look honestly at the clinical paradigm right now as it stands, you basically have two technologies that have really crushed it in the liver in patients. They're safe, well tolerated. They're going to likely be approved. And then you have the rest of the body. And right now, there aren't any real clinical trials that have shown significant progress in non-liver delivery. I think if you've studied drug delivery, this may not surprise you, because if you do an IV injection, everything just kind of tends to get soaked up by the liver. So the idea that we can do non-liver delivery, the fact that non-liver delivery is challenging shouldn't really be a surprise. But this is the reality that I think we deal with. And for that reason, my lab specifically focuses on tools to discover drug delivery vehicles that deliver RNAs outside of the liver. So we focus on non-liver delivery. All right, so just as a brief introduction to what I've done, I focus on endothelial cells historically. You're going to see endothelial cells kind of throughout the talk. But historically, I've developed nanoparticles that deliver siRNAs and other RNAs, microRNAs, sgRNAs, and now mRNAs to endothelial cells. And we've been able to do a few cool things with them, which I'll describe here in a second. So the first thing that we've been able to do is we've been able to demonstrate that these particles really do like endothelial cells. This is just some image of a fluorescently tagged particle in red getting soaked up by a mouse's blue vasculature, and that's fine. But at the end of the day, I don't really care about where the particles go. I'm really more interested in where they work. And so these are the types of slides that we're really interested in. And this was really the slide, I think, that kicked off a lot of our initial work. I don't remember when we got these data, maybe 2013 or something. But what these data show is you have an siRNA that's been administered to a mouse intravenously, so just a tail vein injection, and where the siRNA is being carried in a nanoparticle I designed called 7C1. It was a 731st nanoparticle I tested out of a library of 2,500 different compounds, basically. And we'll get back to why that's important in a second. There's no clever name to this. It's a stupid name. Mathematically, I actually don't even think it's right. But anyway, what you see here is just a simple dose response. So as I increase the amount of administered siRNA from a very, very low dose to a very low dose to a pretty low dose to a moderate dose here, you see that the gene expression drops significantly, which is exactly what you would expect. Again, siRNA turns off a gene. So as we give more siRNA drug, you expect the gene to drop. And for those of you in the siRNA field, you may appreciate the fact that these data are actually, I think, pretty good. We're getting 95% silencing at 0.02 milligrams per kilogram, which is a clinically relevant dose. If you look at the nanoparticle that's going to be approved next year, it works at 0.01 milligrams per kilogram and is well tolerated up to about 2 or 3 milligrams per kilogram. And ours was used by that same company, put into a head-to-head competition, and actually had exactly the same therapeutic window. So people started getting pretty interested in this compound because you can do this kind of low-dose silencing. So I can give a low dose of siRNA, and I can get robust gene silencing in endothelial cells. This is the first time this has been reported outside the liver. So when you get low-dose silencing, you can do a few cool things. I'm just going to highlight one or two. The first thing you can do is, you think about it this way, the nanoparticle is carrying an siRNA, and that siRNA has a sequence. It has a sequence A, which will turn off gene A, right? If I want to turn off gene B, all I have to do is put in a different siRNA, just a different RNA sequence. So I can turn off gene B very easily. So what does that mean? If I can turn off gene A, and then separately I can turn off gene B, it means I can just put the two siRNAs in the same syringe and put them in the same particle and turn off A and B at the same time. So you just have to trust me. We have about 15 papers of data for this, but we don't have time to go over any of that. This is a cross-sectional image of a nanoparticle that's carrying one siRNA. These are scaled the same way. And then this is an image of a nanoparticle that's carrying 5 different siRNAs at once. And what you can see is what you would expect, which is that the particle doesn't really care what the siRNA sequence is, so you can put in 5 different siRNAs all at once. So why is it interesting? Well, as we showed in this Science Translational Medicine paper, and then we have another one that we got in Cell and PNAS and some other journals, and we have 4 or 5 now that are going to be published. In addition, biologists are really interested in multi-gene silencing. Because if you want to make a knockout mouse as an endothelial-specific knockout mouse and you have 4 genes that you want to knock out, good luck to you. That's going to take 5 years to cross and do all that breeding. What we can do is we can just put all 5 particles into the, or excuse me, all 5 siRNAs in the same particle, inject, and turn off all 5 genes simultaneously. And that's exactly what we did in this STM paper. We did 5 genes at once, targeting the leukocyte adhesion cascade in atherosclerosis, which is what this image, which is not to scale, by the way, not to scale at all, is meant to show. There's supposed to be 5 different colors of SI in there. I don't know if you can see it or not. Also, our particles don't magically ghost into cells. And also, these particles are 50 nanometers and cells aren't 20 nanometers wide. But that's OK. All right. So one thing that we take great pride in the lab is we try to develop technologies that other people can use in real life. And there were about 6 months of my life I spent as a part of this original Nature Nano paper that didn't even make into the supplement of that Nature Nano paper, where we optimized every single aspect of how to make these particles. The microfluidics machine we're using, we have 2 syringes, so the relative flow rates, the size of the syringes, the absolute flow rates, everything you can imagine we tried to optimize in order to achieve 2 goals: make the nanoparticles stable so I could ship them to Yale or to Colorado or to whoever's going to use them, and they wouldn't crash out during shipping, and to make them consistent so that if I made a batch of particles on Monday and it came out to 45 nanometers, if I made a batch of particles on Tuesday, it would also be 45 nanometers, not 85 nanometers. So you're getting a consistent batch to batch to batch, and it was stable over time. And basically, it took about 6 months of optimizing, but we did it. And I'm happy that we did it because since that time, since the original paper came out in 2014, something like 20 labs across the United States have used this to do in vivo gene silencing. And it's worked in the hands of every lab that has tried to use it so far, which is good. I feel very happy about that. So these are just some of the publications that have been published to date using the technology, and there are several more. All right, so that's what I've been doing. I've been sort of developing nanoparticles for non-liver delivery, focusing on endothelial cells. And I think that's moderately interesting. That's fine. But I want to get into the stuff that we're doing now at Georgia Tech because I find that infinitely more interesting. And I just want to highlight, and again, I didn't put this in here because Corey introduced me. I put this in every talk I give. I really want to highlight the quality of our students. And I really want to highlight, in particular, 2 students, Corey and Kalina, that have really generated a pile of data that you're going to see here within their first year as grad students. They've been hyper productive. I'm very proud of them, very happy that they're in the lab. But this is, I think, the most important slide in the deck besides the acknowledgement slide. I just want to really highlight Corey and Kalina for their excellent work. All right, so here's I think, the interesting thing that we're going to be talking about today. So please bear with me here. This is important. This is how I learned to discover nanoparticles, and this is how many people in the field discover nanoparticles. This is not the only way to discover nanoparticles. However, many, many, many labs discover nanoparticles this way. This is how you do it. You synthesize a lot of nanoparticles. I can make this chemical structure, or that chemical structure, or that chemical structure, or whatever. I made about 2,500 during my Ph.D., but I'm not even a great chemist at all. There are much better chemists out there that can make 10,000, like Dan Siegward at UT Southwestern, or Yijiao Dong at Ohio State, who can make lipopeptides that are unbelievably good, or Avi Schroeder, Chris Alabi, Kate Whitehead. All these people can synthesize stuff. Making the different nanoparticles is not the problem. The problem is that when we test these things, no matter how many particles we make, and no matter what the chemical structure of that particle is, we are always testing them for systemic delivery by pipetting them onto cells in a dish. So we do, for instance, in my paper, the original paper, I made 2,500 compounds. I screened all of them in HeLa cells, which is standard because it's the only cell line you can expand out to do a 2,500 nanoparticle screen with bioreplicates. I sprayed them onto some HeLa cells. I took the top 3 nanoparticles from that in vitro screen. I injected it into a mouse. I basically prayed that the thing worked. And then we found 7C1, that particle that did work, and then we characterized it in the ways that we just described: tox, biodistribution, low dose delivery, dose response, multi-gene, blah, 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 blah. So this is the workflow that everybody uses. And the key is that we can make, in my little lab, we could easily formulate in a year probably 10,000 particles in a year in my little lab. In Dan Anderson's lab, which is the lab I came from, where they have 60 people working on this problem, they could do 100,000. This is what one of those nanoparticle libraries looks like. Each one of these vials is a different material. And there are a number of different chemistries that you can use to expand out this chemical space. You can use epoxide chemistry, methacrylate chemistry, acrylate chemistry, thiol chemistry, blah, blah. There are tons of chemistries. I mean, MG Finn's in the audience. MG could probably list 50 chemistries off the top of his head that we could use to make nanoparticles. This is what these things look like, though. We can make massive libraries quickly. And the thing I want to point out before I move forward is that for each one of these materials, when we actually turn this material into a nanoparticle, we include other stuff. We put PEG on the nanoparticle. We might put some cholesterol on the nanoparticle. The PEGs can have different molecular weights and different lipid tails. And for each one of these materials that we can synthesize, and again, you can synthesize up to 10,000 if you're Dan Siegert, you can probably, as an honest scientist, formulate 500 variations off of that material by adding this cholesterol or that PEG or DSPC or DOPE or whatever. That's a conservative estimate. Okay, so we're getting into the millions and tens of millions of compounds that you could honestly ask as a scientist, hey, is this going to go to the liver, or is it going to go away from the liver, is it going to go to the spleen, is it going to deliver RNA to the heart, whatever. Okay, so after we synthesize these things, what we're trying to do, let's say that we're focusing on heart and lung delivery. I'm trying to predict whether a nanoparticle I inject into the vein of a mouse is going to avoid the liver, avoid the kidney, avoid the spleen, avoid the circulating immune cells, get into the lung or the heart, depending on which one I want, get into the right cell type in that very complicated tissue microenvironment. All of this is happening with a protein corona. All of this is happening with different pulsatile blood flows. If you're in the arteries, you're getting pulsed. If you're in the veins, you're kind of dribbling around a little bit. And the way that we're trying to do this is I'm spraying it onto HeLa cells in vitro. Now, this is really, really important. I'm not bashing in vitro in general. I want to be hyper clear about this. I'm not bashing in vitro in general. That'd be idiotic. What I'm saying is that if this is the output that you're looking for, a systemically administered particle, and you want to really identify which particle has the best chance of working after systemic administration, this may not be the best model. In vitro is going to be awesome for a lot of other stuff, but if you're trying to model something that involves all of the organs and all of these components that all talk to one another, it's going to be hard to do that by pipetting them onto HeLa cells. That's what I'm saying. I'm not bashing in vitro in general. Okay, so we have this problem. We can synthesize, let's say, 2,000 compounds, and just for conservative numbers, let's say we can make 2,000 compounds, and I want to test them all in vivo. That means I can either run an 8,000 mouse experiment and go bankrupt and have no grad students willing to work for me. So I can do a lot of mice, or I can do a lot of compounds per mouse. And the only way we figured out to afford to do this was basically, how can we do a lot of compounds per mouse? And this is where I think my background from the Broad Institute kicked in, because it seemed like a good idea to do some barcoding stuff. So what we can do is we can basically, now we can barcode nanoparticles. In this arrow here, this high throughput nanoparticle formulation, is just an arrow, but that's where that 6 months of work went in. Because during that 6 months when I was a grad student, I optimized this arrow. And so now we can make a lot of different nanoparticles in a single day. So this is what it actually looks like. You can take, I'm going to flip back one. You can take basically a DNA barcode, which, by the way, for those of, many of you actually may not know what that is. A DNA barcode is simply a DNA sequence that we know. I just know what this sequence is. That's all it is. And we can design this DNA sequence in different ways. We can make it big or small. We can make it form a hairpin, which we did by accident, which is horrible. Or you can unhairpin it. You can do a lot of stuff. You can do all these DNA barcode designs. Oh, and shout out to Anton, by the way, for figuring out what was wrong with our system. Anton saved me probably tenure. I think Anton saved me tenure if I get it. By not, because we had no idea what was going on a little bit earlier this year. So thanks to Anton. So thank you, Anton. All right, so we have a DNA barcode that you can rationally design. We have material, and we have PEG here. And we can basically formulate these particles so that they're not carrying siRNA, but instead they're carrying DNA barcodes instead. Very easy. And in a single day, we can make a lot of these particles. So that nanoparticle 1, which would be this yellow nanoparticle, carries DNA barcode 1, which would be this red barcode in this cartoon. The second nanoparticle, which is a totally different material, would be this gray material with this other barcode, and so on. And we can actually do this not 3 times in a day, but I don't want to lie, what's the max we've done? 158. Made? Yeah. No, we've made more than that. In a day, we analyzed 158. Anyway, we can do, I think, up to about 200 to 250. We can make 200 to 250 particles in a day. We have 3 stations, and we can get to about 250 in a day. We then do a very hard quality control. So this arrow also is non-trivial, where we basically make sure the particles that we make are stable and they're small and they aren't going to mix together. And so we can pool, so we take these, we make a bunch of particles, each one has its own barcode. We pool them together and then we can administer them to whatever you like. We haven't done primates yet, it's something I'm interested in doing. But right now we've done a bunch of mice. You can do a normal mouse or knockout mouse, as we'll see later. You can do any animal model you want. You can do primary cells, you can do cell lines. And then you can basically take the DNA from the cells, the primary cells, or from the animal, and you can just deep sequence to see which nanoparticle is where. So this is a nanoparticle targeting heat maps, actual data we've generated. And I think in this, my guess is, I don't remember what, but this is probably cell types on the x-axis, and this is probably nanoparticles on the y-axis. So that we've done up to, I think the biggest data set we generated is something like 30 cell types times 150 nanoparticles. And for those of you who've done, all in vivo, and for those of you who've done nanoparticle stuff, you know that that's just a massive scale. I'm used to doing 1 by 1, not 150 by 30. So these are the data that you're going to see. These are called normalized delivery. Has anybody here done RNA-seq? If you've done RNA-seq, this is equivalent to that RNA-seq, like counts per million. It's the same thing. We basically take the total number of DNA counts from a tissue. So this could be, for instance, in vitro endothelial cells, or this could be, just call this long 1 for simplicity. This is just long 1. And let's say we got 6 DNA reads from long 1. In reality, it's not 6, it's like 10,000, but just for simplicity. And let's say that if we put 3 nanoparticles in there, like in that previous cartoon, and we got 2 reads from each nanoparticle, this would be the normalized delivery. It'd be 33% for each particle. Easy enough, right? It's like a pie chart. Now if you took lung 2 from a different mouse, or heart 1, or whatever, some other sample, and you do the same thing, then, and you got 1 gray, 4 green, and 1 red, then this would be your normalized delivery. You'd have 66% of it would be green. That's what the y-axis is going to be, just a pie chart. So this is a very small piece of data that we generated, but this is one way to illustrate that these data are real. I'm going to start getting into some control experiments, so please stay with me because I like to demonstrate that our stuff is robust and I'm not just making things up. This is one of the control experiments that we've run. This is 104 nanoparticles tested at once. Those are all the black dots here. This is normalized delivery on the y-axis here. So we did this experiment and we said, okay, we're going to do 104 nanoparticles. And we're going to throw in a negative control, something that we know isn't going to work. And that negative control was a naked barcode. And then we're just going to sequence everything, and we're going to see how everything behaves. And what we found is that in this sample, and I'm going to show you, I think, in the next slide, 18 other samples from the same experiment, all 104 nanoparticles, all of them, outperformed the naked barcode. The naked barcode completely fell out. And this happens in every single experiment that we run. We include this negative control in every experiment that we run. So this is the rest of that data set. This is a log 10 plot. There's 104 particles done in 18 tissues, both in vitro and in vivo. And we just highlighted the negative controls here. So in every single sample, the negative control falls out as exactly as you would expect. So, okay, that's a negative control, that's fine. What about positive controls? Because we're going to start comparing stuff in a second. So we want to ask another question, which is, if I want to compare delivery of nanoparticles in a normal mouse and a wild-type mouse, let's say I plot normal mouse on this axis and wild-type mouse on this axis and knockout mouse on this axis, and I want to see how different or similar, either similar or dissimilar they are. I need to be able to say, okay, if they are similar, I get a straight line. They are similar, I get a straight line. If nothing changes in the animal, I get a straight line. So we had to figure out a control, positive control, that should be a straight line. And so what we did was we took the same 104 particles, we administered them in vitro to an endothelial cell line that we got from Hung Jun at 20 nanogram total dose. So 20 nanograms total dose averages out to 0.2 nanograms per particle. And then we took the same batch of particles on the same day, and we administered them to some IMAX sitting the next well over, and we did it at 100 nanograms total dose. And we figured that, hey, the particle that does well in the medium dose here, so it would be far along this x-axis, should also do well in the higher dose. So it should be high here. So we should be up here. If we have a particle that performs poorly in both, like this naked barcode, the negative control, it should be low in the medium dose and also low in the high dose. So it should be here. So if you look along the diagonal, there should be a straight line. in this control experiment, this is exactly what we get. The R squared value is something like 0.999. So we were pretty happy about that. Okay, I always get asked, well, you know, how do you know they're in the cells, which I think is an interesting question because we take them at a time point that where they should be in the cells, but we also image them with Phil Santangelo. So here are some fluorescent barcodes. So, okay. One of the other controls we wanted to run, and this is a control that really was really bothering me, was we wanted to see if the particles are going to mix in solution. So I did this back at MIT. So the way that we did this was we said, okay, we're going to make 4 nanoparticles. We're going to barcode all 4 of them. And we have nanoparticles that we know how they should behave. So we have a liver nanoparticle that's allegedly on the way to the clinic, and then 3 lung particles, one of which is allegedly on the way to the clinic. And we barcode all 4 of them, we inject them into the mouse, and we sequence in the lung, and sure enough, we see that the lung particles outperform the liver particles. But we said, oh, that doesn't tell us anything about mixing. How can we tell these particles are mixing? So we said, okay, what if we take that same batch that we made and we leave it out on the benchtop for a while and we inject new mice the next day? The hypothesis was, well, if these things are mixing together, then if it sticks on the benchtop and we inject some new mice, then this is going to change. So we did that experiment, and this is what we got from the mice we injected the next day, which is very good. So these particles, at least, were not mixing. Now, I'm not going to say that all particles are not mixed. Again, that's silly. But at least we have a control experiment to see how much your particles are mixing together. And for those of you who are saying, well, why do we care about particle mixing? What I'm really worried about is, okay, I'm going to pool 300 particles together or 200 particles together. If I get particle 1 and particle 2 in the same batch, do they form particle 1, 2? And then I'm getting junk in, junk out. Okay? We also wanted to see if barcode sequence affected stuff, and basically it doesn't. The way we did this was we took nanoparticle 1 and formulated it with barcode 1. Nanoparticle 2 got barcode 2. 3, 4, and 5 got 3, 4, 5. And then separately, we formulated nanoparticle 1 with barcode 6. Nanoparticle 2 with barcode 7. 3, 4, and 5 got 8, 9, 10. And we injected all 10 together. And we said, OK, if the barcode sequence is affecting the delivery, then these bars should be different, and they're not. So this is the type of scale that we can get up to now. We validated this system using a bunch of other controls as well. But we're comfortable now to the extent that we can generate very large nanoparticle targeting data sets. So this is, again, a heat map. Red means bad, blue means good. And this is a heat map of normalized delivery. All right, so just a massive normalized delivery data set. And what you see is 104 nanoparticles on the y-axis, and I don't know how many tissues and cell types on the x-axis, a lot. And the thing I want to really highlight here is that this is a paper in my field. So these are the types of papers I'm used to publishing, where this might take me a few weeks to generate these data, and if I do a few in vitro things and follow up with this, then I can get my JCR paper, and that's what I'm accustomed to doing. So we can really generate data at a scale that is new. Okay, the other thing that's really cool about this, and I think this is a more interesting aspect of this technology, is we can ask fundamental questions about delivery that are really difficult to ask otherwise. So for those of you who do delivery, I'm not being controversial here. I think it would be fair to say, at least in my experience, maybe I'm the worst nanoparticle person in the world, maybe the case. But in my honest experience, I would say that most of the stuff I make doesn't work. And if it works, I don't know why. And there are 2 things that leads me to believe. I need to understand which material properties dictate whether the nanoparticle is going to work. That's one outstanding, very simple question that I still think is unanswered. The other question I want to study is which biological pathways dictate delivery in vivo, which is also unclear. Right now, I don't know of any master transcription factors that dictate particle delivery. I bet you they're out there, though. And so we can start to ask these fundamental questions using this technology, because now I can put in 200 particles into this mouse, and if I design my experiment cleverly enough, I can put the same 200 particles into a different mouse and get to some of these questions. So one of the other fundamental questions that we're interested in is sort of the process of nanoparticle discovery. And so I'm just going to highlight this question here. And it's a pretty simple question. At the very beginning, I said, OK, we have 2,500 different nanoparticles. We test them in HeLa cells or whatever. And then we take the top 3 and put into an animal, and we pray that the thing works. And so that led me to ask the question, again, very simple. Does in vitro delivery predict in vivo delivery? To what extent does in vitro delivery predict in vivo delivery? Because people like me have been testing for in vivo delivery using in vitro assays, again, static cell culture assays. I'm not bashing any of the more advanced cell culture stuff. I'm not bashing microfluidics. I'm not bashing organoids. Just my field's been using static cell culture. We've been doing this for 20 years. Okay, so the way that we did this was we took a bunch of particles and we administered them. We took the same batch of particles and we administered them onto cells in vitro. Again, static cell culture. So I'm not talking pulsatile stuff. A lot of people in this room do very fancy in vitro stuff. I'm not talking about that. I'm talking about what my field does, which is static cell culture. You put it onto the cells in culture. We took the same particles on the same day and we put them into the mouse. And then we did this with macrophage cells and endothelial cells. And we did that because we can isolate macrophage and endothelial cells from the mouse. So I take the same pool of particles, I dump it onto some endothelial cells in a dish, I put it into a mouse, and then at the same time, I get the DNA from the cells and I fax sort endothelial cells from the mouse. So I'm directly comparing endothelial cells to endothelial cells in vitro to in vivo. And we would reason that if this relationship is a straight line, if we get an R-squared value of something high, then in vitro would predict the in vivo delivery. And if the R-squared value is very low and you see this kind of scattery plot, then in vitro delivery does not predict in vivo delivery. Ta-da! This was terrifying. We've repeated this, I don't know how many times, a bajillion times. This is what we get every single time. This is a collected data from 400 and something, or no, 300 particles or something done over multiple experiments. The R-squared value between the thing that we've been trying to use to predict in vivo delivery and in vivo delivery averages out to 0.03, which is basically throwing stuff at a board. Okay? This is the endothelial cell data. Some of you might be saying, oh, okay, well, did you vary time point? Did you vary cell type? Did you vary cell culture conditions? Yes, yes, and yes. Our supplementary figure in this is figure 2B through D in our paper that we're submitting right now. Our supplementary figure goes A through OO. Okay, and many of, like Z is actually 4 panels. So we have like 100 data sets that show this. This is the same thing for macrophages. This is also multiple cell types. So what you can see here is, and I'm going to flip back here. It's the same thing here. This is endothelial cell in vitro against heart endothelial cell in vitro versus lung endothelial cell in vitro versus marrow endothelial cell. All right? And we have the same thing for macrophages as well. That's the reason we did endothelial cells and macrophages is they're in every tissue, so I can isolate them from every tissue, because we wanted to make sure that any effect we were reporting wasn't tissue specific. So what this means for us is that when I think about screening nanoparticles, it means I either have to work with somebody who's doing organoids or microfluidics, or I have to do it in vivo. The static culture stuff that I've been doing since 2009, as far as we can tell, does not predict what I'm trying to get it to predict. And just as a reminder, this is what that straight line looks like. Again, this is why we ran the positive control experiment. If you have something that should predict something else, in other words, dose A and dose B of the same stuff in the same cell type, you get an R-squared value of 0.99. All right, so we were able to do some sort of statistical analyses and ask, okay, well, if you take the top nanoparticles from the in vitro screen, do they predict in vivo delivery? And again, the answer is more or less no. Sometimes yes, but more or less no. So we can run all sorts of statistics. This is one very simple. We did a bunch of analyses, but this is just our most simple analysis to understand, I think. This is basically how well did this particle do in vitro. So this is we did the in vitro screen. This is the best particle in vitro, second best particle in vitro, third, fourth, and fifth best particle in vitro. And then we said, okay, well, how well does that particle do in the corresponding in vivo screen? And what you see is, encouragingly, you do see some particles that do pretty well in vivo. So this nanoparticle here did well in vitro and was also #1 in vivo, which is good. This nanoparticle was #1 in vitro and was probably #3 or something in vivo, which is pretty good. But you also get this scatter up here. So your best performing particles in vitro aren't necessarily going to be your best performing particles in vivo. Yeah. And then finally, we also wanted to ask a very simple question, which is, if I take 3, 5, or my top 3, my top 5, or my top 20 nanoparticles from an in vitro screen, and I just ask the question, what are the odds I get my top nanoparticle, my top 2 nanoparticles, and my top 3 nanoparticles from in vivo? What are those odds? And basically what we find is that the odds of taking, if you take 3 nanoparticles from your in vitro screen, you have about 11% chance of getting your top nanoparticle in vivo. You have a 22% chance of getting, you have a 22% chance if you go up taking your top 5 nanoparticles, And if you take your top 20 nanoparticles from your vitro screen, you have about a 45% chance of getting your top particle in vivo. And you can see how this kind of drops off if you're trying to get your top 1, 2, or 3. Okay, so we can ask questions like that, which I think are kind of cool. Great. The other question that I wanted to ask is how are different cell types targeted in vivo? And what I mean by this is when we do biodistribution in an animal, what we do is, we all do this, I do it as well, we take a fluorescent dye, we inject a mouse, and we either do an ex vivo imaging or whatever, and we say, okay, there's such and such fluorescence in the spleen and such fluorescence in the liver. And some people also will go in and look at cell type specific fluorescence, which I think is a little bit more interesting. But we wanted to be able to look at cell type specific delivery within a complex microenvironment for many particles at once. And luckily for us, it's very, very easy to do. Because we can just do this. Take the barcoded particles, administer them to a mouse, sack the mouse, we focus on the spleen here. And I want to thank Lena for helping us learn how to do spleen, as well as Rafi Ahmed for teaching us how to do this stuff. And then what we can do is we can actually fact sort out different cell types from the spleen. So these are the stains we got from Rafi Ahmed over at Emory. And there's just a lot of stains. But basically, you can isolate B cells, endothelial cells, and blah, blah, blah from the same spleen. And then you can measure delivery in all those different tissue beds. And you can form a little heat map, which I think is kind of cool. So this is the heat map from the spleen. This is, I don't remember how many particles this is, some amount of particles. And I want to point out a few things here. One thing that we have seen over and over and over is that this is macrophages in vitro. We did the same experiment here. When we took these particles, we also administered them to some macrophages in vitro. We always do this. And what you see is that this is unbiased clustering. The in vitro macrophages cluster totally distinctly from all the in vivo samples, as we would expect. And again, they cluster very far from the macrophages, which they're supposed to be predicting. The other thing we noticed, and we haven't done this experiment, we haven't repeated this experiment to look for this relationship specifically, but I forget who. Was it somebody from the Kwong lab or somebody from our lab? I don't remember if it was Corey or somebody from the Kwong lab. Somebody noticed that these cell types are dividing into different lineages. Now, we haven't seen this repeated. We haven't repeated this experiment to see if this is true. But they said that, hey, very interestingly, the two types of dendritic cells that we isolated actually clustered together, which is kind of cool. And they also clustered with the other phagocytic cells, which all come from the same progenitor, to my understanding. I'm not an immunologist. So these clustered together more closely than the lymphocyte lineages, which I thought was kind of cool. Who knows if that actually is real? I don't know. This is an internal thing, so just saying. But I'm on record, so yikes. Okay. For the record, I don't know if that's true. Okay, good. The other thing I want to point out is that we try to, this high throughput in vivo screening stuff, you need to be able to validate these hits. So I'm not going to sit up here and say that like every in vivo screen is going to be exactly right and particles are never going to mix. Again, that's stupid. What we think is that the in vivo screening is a useful tool to get us to a good top 10 list, a better top 10 list than the in vitro screen. But once you get to that top 10 list, you have to validate that what you're seeing is real. So we had to do that for our data set. So I don't know if I highlighted it. Ah, crap, I didn't. One of these particles, I forget which one, is what we call LNP1. It basically did really well. It was very, very blue in the spleen. And one of them was LNP2. It did really poorly in the spleen. It was like very red in the spleen. So we said, okay, we want to make sure that these spleen data line up with reality when we test LNP1 and LNP2 separately from one another. So we want to compare our pooled stuff to our individual stuff. So what we did is we took LNP1 and we took LNP2. This is just the data from the spleen from the high throughput thing. You can see that LNP1 is really outperforming LNP2 in every single cell type in the spleen. Don't worry about this. And then what we did is we took LNP1 and formulated it with a fluorescent barcode. And we took LNP2, we formulated with a fluorescent barcode, and we injected separate mice with them. And then we waited and we took the spleen out and we looked at fluorescence like we would do normally and traditionally as a drug delivery people. And this is what we saw. We saw it was actually perfectly recapitulated. So LNP1 showed up a lot in the spleen. LNP2 didn't show up in the spleen, just as we saw in the high throughput in vivo screen. This is the ratio. So this is, whatever that is, like 15 or something. All that is this divided by that. So red divided by blue equals this. we're plotting this because this is that fluorescence ratio as well. So we were within that range, so we were pretty happy about this. Yeah, so the only point I'm trying to make here is that this high throughput screening thing is not perfect, and just like all high throughput screens, you have to validate your hits. We can do some other cool stuff, so we can study pharmacokinetics and all sorts of things. So if you can think of a cool experiment, please let us know. We're trying to do our best to try to find cool experiments to do. This is one of the experiments where you can inject, in this case, 30 particles, and we only show 10 here because it gets messy. But you can do 30 particles and stack the mice at 15 minutes, an hour, 2 hours, 4 hours, 8 hours, or 24 hours and see different pharmacokinetic things. And then use that to calculate area under the curve in a high throughput way. Yeah. And then we can also look at optimizing tissue delivery, because what we have here is an opportunity to not look at just one cell type or tissue type, but actually look at a bunch of cell types and tissue types and try to find particles that are, for instance, very good in heart and pretty bad in liver, or very good in heart and bad in liver, kidney, spleen, or whatever. We have some other data, but I'm reaching about 45 minutes or so, and everybody has stuff to do, so I'm going to kind of end it there. I want to thank everybody in my lab. Many of them are here today, and everybody makes an important contribution to this work. So I think I have great lab members. I'm very lucky to have those lab members. This has changed to Zubao Gan, who's going to be joining us in a few weeks. He's a chemist, and we're looking to expand our chemical space. I'm really excited to bring him on board. And then I also want to thank the funding sources that have allowed my lab to grow pretty quickly since I've been here. Yeah, so this is the second most important slide. And great timing, Kalina. Kalina is in the back. This is just re-emphasizing the most important slide, which is that Kalina and Corey have really done a fantastic job on this work. Great. So with that, I'll take any questions. And if you guys have if you want to collaborate, or you want to talk about this technology and try to understand it a bit, or say, oh, I think that's crap, you should be doing this or whatever, let us know. We're right at the beginning of this, and it's a very exciting time. We've developed 2 other screening systems haven't had the chance to talk about. One is, so this screening system we just talked about is named Jordan. It's named after my wife. Joint Rapid DNA Analysis of Nanoparticles. Kind of a stretch, but whatever. And then we have a second system that we call Find. This system tells you where the particles go. Find tells you where the particles work. So we have a functional high throughput barcoding screening system as well that's under review right now. And then we have a new system that's almost up and running where we can not only tell where the particles go, but we can get that pie chart that we talked about. We can also get the size of the pie. So we have 3 systems up and running. We're starting to get into these very cool studies where we'll put in 200 particles into a wild type mouse, and then the same 200 particles into 5 different knockout mice on the same pathway to see which genes affect delivery. We're right at the beginning of this, and it's a very exciting time in the lab. So if you are interested in learning more or just want to say hi, please feel free to contact us, and I'll take any questions. Yeah, Brandon. Yeah. Yeah.
Male versus female, or old versus young,
yeah.
How much does which mouse I pick?
Yeah. It's a great question. So it's a great 2 questions. So yeah, you're right on point. We think about this stuff. We've done a bunch of different cell lines as part of our kind of confirmation studies, that supplementary to A through 00 thing. We had all these different cell types and stuff. And the cell lines are very predictive of one another. So whether you're talking about macrophage or primary or whatever, they're very predictive of one another. The R-squared values are high, which in my mind makes sense. You're dumping stuff onto cells, and there are going to be some differences, but they're not very dramatic. For the mouse stuff, we thought about this. My dream is to publish I already know the title of the paper, by the way. I already know the title of the paper. The title of the paper is going to be Nanoparticle Drug Delivery is or is not conserved. And we're just going to do 10 different mice, 5 different rats, a monkey, and a pig, and just see what happens. That's something that I'm really interested in doing, because one of the key questions, not to be coy, one of the key questions, and you're alluding to this, is when you're talking about the clinical translation of these things, you can go into industry, you go into the most well-funded pharma company in the world. If they're doing nanoparticle stuff, they have no idea if that mouse is predictive. No idea at all. Same thing with virus people, right? That's why Mark Kay has gotten so much press when he did his FRG mouse study. So we're trying to figure out how to make inroads into that question, which is how on earth do you actually predict human delivery? We don't know the answer to that yet, but we're trying to figure that out. Yeah, that's a great set of questions. Yeah, Todd.
James, thank you very much. How deeply have you gone into the material science aspect of the particles to see if you can sort of make predictions about why certain particles with certain locations?
Yeah, so it's another great question. We have done very JV analysis because we don't know how to do this multi-component stuff. So what we'll do is we'll be like, peg size, like small, medium, large, and here are like 40, 40, and 50, and like from this screen, and is there any relationship? And we see no, and stuff like this. I'm trying to figure out, I'm trying to find people at tech to work with to do this multi-component analysis because it's not going to be one variable. Anytime we plot one variable, we don't see any strong relationship. So as an example, when we plot size, so we can plot delivery against size because we record the sizes for every particle we've made, and we've probably done this for 1,000 particles now. When you plot delivery versus size for any tissue so far, there's no relationship. Now that doesn't mean that size is irrelevant for all particles. It could be that size matters for other particles, and that could definitely be the case. But in our experience with these LNPs, if you look at 1,000 LNPs, there hasn't been a relationship between size and delivery. So there's always going to be this multi-component thing. And to be honest, we need to become better mathematicians to figure out how to address those problems. Yep.
With regard to, so you just mentioned trying to find some translation into like human
Yep.
patients. How large of a tissue sample size do you technically need to be able to analyze the distribution of your particles? Like, could you take a biopsy? Is it possible that you could just get a sense of the delivery of things in humans just by using biopsy?
Yeah, I think we could. The only issue is we never be able to inject humans with not with that attitude, right? Yeah, but one thing that we are interested in, there's this guy at NCI that I had a chance to meet when I was in DC, and he is actually doing these excisions of, he's a surgeon, and he takes out tumor nodules from the liver and he perfuses them to look at transcriptomics. He does all these studies on these nodules that he basically keeps alive in like some chamber thing. And he and I are trying to get money now. I don't know how we're gonna get the money, but what I really want to do is like hook up our particles into this chamber thing and take these biopsies. You take 10 different nodules from the same patient and then have healthy tissue from that same patient, put the same pool of particles in and just measure. I think that'd be super interesting. But the key is that we have to be able to perfuse them in a realistic way. You got to hook them up to a blood supply, basically. Does that make sense? It's a great question. Yep. Yeah.
Sort of a follow-up to that question. Do you understand the distribution within certain organs of your particles, or are you not concerned about that?
Yeah, to be fully candid again internally here, I don't care about the tissue level distribution. Fully candid. The limitation to, I'm not saying that as a general rule, that I don't care about tissue. I mean for our assay, if I take an entire lung out and I sequence it, that means I'm either sequencing stuff that's in lung cells or ECM. And statistically, I'm probably going to be measuring ECM just because it's harder to get into a cell than not get into a cell. So for that reason, we're very facts heavy. So Corey and Kalina and Melissa and everybody in my lab is very, very good at doing facts. We always take the organs out and we'll fact sort simply because I don't know if I'm measuring ECM or not. Again, it's not bashing whole tissue stuff in general. Just for us, I don't know how to interpret those data, if that makes sense. Hey, Scott. Yeah.
You made the comment about the sort of hierarchy of animal experiments and predicting one animal to the other. What about even going back and saying, you're doing static, two-dimensional culture. What about three-dimensional culture, diffused organ culture, predicting mice and predicting the biggest primates, etc.?
I think it's a great, I think it's a killer idea for a few reasons. And yeah, just great idea. What I think this presents us with a nice opportunity to have a positive control experiment for instance, a microfluidics person. So say a microfluidics person, and like Tony Kim's a perfect example of this. he makes these fantastic microfluidic devices there that are meant to recapitulate brain delivery, right? It makes sense to me that we could use the in vivo data as a positive control. So you could be a microfluidics person. You seed your device with a bunch of mouse cells at first, and we compare different devices, device A, device B, device C, to the mouse. And then we say, oh man, device B is really killing it. Device A and C just aren't getting it done. And you can actually figure out how to tune the device and then seed that device with human cells. And then go into the animals and do that same sort of thing for larger animals. The limitation for us is that I don't know how to, I don't know how to work with PDMS. It'd probably catch on fire if I touched it. So we don't know how to do any of the more complex tissue stuff. It's a very simple limitation. We just don't know how to do it. Hey, yes.
Can I ask just a quick question about quantitation? So
Sure.
if you know that you inject a bunch of particles and you get relative distribution in tissues, any sense, for example, on the lung example you just talked about, any sense of what the overall mass of, what percentage of stuff you inject
No.
do you get where you're measuring?
No, and that's really annoying. And that's what, so we have this ddPCR-based barcode that we have started to develop that gives us both pie chart and size of pie. And that's exactly what that third system is, because this system had two limitations in it. One was it wasn't functional data, it was just distribution. Second was I don't have size of the pie, I just have the pie. And we were really discouraged by that because it means that we can't compare across experiments. And you can imagine if you have a wild type mouse and a knockout mouse and you have the ddPCR capability or the absolute pi capability, then you can get into some really interesting studies that are kind of obvious to think about, and that's where we're moving. All right, well, thank you guys very much and have a good rest of your day.